Welcome to our presentation. We are the team number 4, which is composed by Devani Len, Serena Montserrat, Erika Fernanda, Valeria Villarreal Piña, Luz Estefania Cruz Méndez, and me, American Sua. The topic of this stage is obtaining energy, and the topic that we are going to talk about in this presentation is photosynthesis, cellular respiration, and fermentation.
First, we're going to talk about the photosynthesis. The photosynthesis is the formation of chemical energy stored in the glucose bonds and oxygen releasing in the presence of sunlight. In other words, it's the process by which plants use sunlight, water, and carbon dioxide to create oxygen and energy in the form of sugar. In here, we have the formula. We have 4 stages in the photosynthesis. That is the absorption of light. The second is the electron transfer leading to the reduction of NADP plus to NADPH. The third one is the generation of ATP that I'm going to talk about that later. The fourth one is the conversion of carbon dioxide into carbohydrates. This is the carbon fixation. Talking about the proteins, 4 major membrane protein complexes in the photosynthesis are the photosystem 1, the cytochrome B6F, the photosystem 2, and the ATP synthase. Glucose combines with oxygen, that is called oxidation, forming carbon dioxide, water, and 38 molecules of ATP.
And now the following topic is cellular respiration. The definition is that it is a series of metabolic processes that take place within a cell in which the biochemical energy is harvested from organic substances, for example glucose, and then stored in energy carrying biomolecule, which is ATP, for use in energy requiring activities of the cell. The equation of this process is the reactants are C6H12O6 plus 6O2, and the products are 6CO2 plus 6H2O.
In the cellular respiration, we have the catabolism. The pathways responsible for breaking the different compounds are known as catabolic. Therefore, the phase in which they are carried out is known as catabolism. The catabolism has 3 stages. The first one is where the macromolecules are digested into smaller molecules. For example, the starch. The starch is digested into glucose, sorry. The triglycerols are digested into fatty acids and glycerol, and the proteins are digested into amino acids. The second stage is where the molecules obtained in the first stage are transformed into acetyl-CoA. For example, the glucose and the fatty acids are the ones that are transformed into acetyl-CoA. The third stage and the last one is where the acetyl-CoA enters into the Krebs cycle. We are going to talk about this Krebs cycle later. In this one, the addition of CO2, the reducing equivalents that are used in the electron transport chain are generated. Here we have the equation where acetyl-CoA transforms into CO2 plus H2O. Now we have the anabolic. The anabolic basically is the opposite of the first stage of the catabolism. The anabolic pathways are those responsible for synthesizing from simple molecules to other more complex one, which sometimes function as an energy store. Starch in plants and glycogen in animals, triacylglycerols, or structural as proteins. As I said before, the anabolic is the opposite of the first stage of the catabolism. So the glucose is synthesized into starch or glycogen. The fatty acids and the glycerol is synthesized into triacylglycerols, and the amino acids is synthesized as well into proteins.
Now we will explain the Krebs cycle, which is a sequence of reactions by which most living cells generate energy during the process of aerobic respiration. It takes place in the mitochondria, consuming oxygen, producing carbon dioxide and water as waste products, and converting ADP to energy-rich ATP. The first step is when glycolysis, the pathway that glucose catabolism performs, takes place, and it is aerobic. This pathway takes place in the cellular cystosol. There we have the equation that represents this first step. Also in this step, the decarboxylation reaction of pyruvate is included, even if it takes place in the mitochondria by a multi-enzyme complex called pyruvate dehydrogenase. Okay, now in the step 2, acetyl-CoA will be used in this cycle, in which most of the reducing equivalents that will be used in the electron transport chains are generated. There again we have the reaction that represents this step. And finally, in the step 3, in here we already have the reducing equivalents to fit the electron transport chain, which will give H2O as the final product, which results in the synthesis of ATP. Here again, we have the general reaction of the cellular respiration using glucose as substrates.
Next, we are going to talk about fermentation. Fermentation is a chemical process by which molecules such as glucose are broken down anaerobically. The route by which glucose is catalyzed or broken is glycolysis, and the equation consists of 1 mole of glucose turning into 2 moles of ethanol and 2 moles of carbon dioxide, producing 2 moles of ATP in the process. We have 2 types of fermentation. First, there's lactic acid fermentation. It is a biological process by which glucose, fructose, and sucrose are converted into cellular energy, and the metabolite lactate. It is an anaerobic fermentation reaction that occurs in some bacteria and animal cells, such as muscle cells, and if oxygen is present in the cell, many organisms will bypass fermentation and undergo cellular respiration. However, facultative anaerobic organisms will both ferment and undergo respiration in the presence of oxygen. It has 2 steps. It is glycolysis and NADH regeneration. During glycolysis, 1 glucose molecule is converted into pyruvate molecules, producing 2 net ATP and 2 NADH. In NADH regeneration, the 2 NADH donate electrons and nitrogen atoms to the 2 pyruvate molecules, producing 2 lactate molecules and regenerating NAD+.
Well, now we will talk about the next type of fermentation, which is called alcoholic fermentation. Alcoholic fermentation, also referred to as ethanol fermentation, is a biological process by which sugar is converted into alcohol and carbon dioxide. Yeast are responsible for this process, and oxygen is not necessary, which means that alcoholic fermentation is an anaerobic process. Alcoholic fermentation is commonly used for the production of alcoholic beverage, such as wine and beer, of ethanol, fuel, and of bread. Also, it is used by some fish for energy production in situations of lack of oxygen. Alcohol fermentation involves the conversion of pyruvate into ethanol and carbon dioxide. It is a 2-step process. In the first step, glucose is converted to 2 pyruvate molecules by glycolysis. Also, a carboxyl group is removed from pyruvate and released in as carbon dioxide, producing a two-carbon molecule called acetaldehyde. In the second step, NADH passes into its electrons to acetaldehyde pyruvate. It is converted to ethanol and carbon dioxide by using a molecule of NADH. And in addition, here we can see the process step by step in this diagram. Well, this is the end of the presentation.